Welcome to the first UU Forum for March 1. It's my pleasure and honor to introduce to you a man who really needs no introduction because he's been a member of the church since 2003. Is this coming?
It's a newcomer.
A newcomer. Oh, okay. I see. Okay. A newbie, yes. He's been involved in discovery and development of disease therapies for over 30 years. He has a BS in chemistry from Georgetown University, a PhD in pharmacology from the University of Virginia, and a postdoctoral fellowship at Harvard Medical School. He's worked for large and small drug development organizations in a variety of therapeutic areas. From 2009 to 2022, he worked on developing a treatment for Alzheimer's disease at a startup pharmaceutical company here in Pittsburgh, where he served as executive director of pharmacology. Nick will talk about what Alzheimer's disease is and what its causes are thought to be. He will go over recently approved therapies and their effectiveness, and will touch on the current drug development pipeline and what types of approaches they represent. Lastly, he will give an overview of the drug he helped put into clinical trials and how it is thought to work. Pay attention because there's going to be a quiz at the end of the talk.
Thank you, Clint, and thank you, Maria, for inviting me and the rest of the team. Back, I actually gave a talk on Alzheimer's disease and some of the research going on in the area 15 years ago. And that was just after I had been working in the area for about 2 years. So I don't know what I was trying to do talking about it. But since then, I've learned an awful lot about Alzheimer's and its treatment. I've learned enough to know that I know very little. And I've also, being at this congregation for a long time, I know that whatever I might think I know a lot about, there's probably somebody in the audience who knows more about it than I do. So please bear with me and be gentle, and please be free to add what you want, especially at the end. I think we probably all know what Alzheimer's is. We may know somebody close to us who has Alzheimer's disease. But just to briefly say, it's the most common cause of dementia in the world, and in elderly people. There's currently estimated to be 7.2 million cases of dementia in the U.S. When I gave this talk last time, my notes said 5.2 million, so you can see what's happening. 30 million in the world, and 60% of those are estimated to be Alzheimer's disease, and the rest other types of dementia that do occur. Overall, the general population in the U.S., the risk of getting Alzheimer's disease at some point is 10%. The biggest risk factor for the disease is age. But there are also inherited factors that affect the incidence of Alzheimer's and the age of onset. I think we know a lot about this, probably the loss of memory, first thing that happens, inability to learn new things, loss of language function, and a progression of failure of brain functions. There can be also some psychological issues such as depression and psychosis that occur late on. In some people, not in all. The manifestations of the disease can vary a lot from person to person. And the disease is progressive and fatal. So when I talk about the progression in years, this kind of gives an idea. Normal aging, we all go through some loss of our cognitive abilities and ability to memorize things. It probably starts after we're a little child. If you've ever played any memorization games with small child, you know that they have, like a 3-year-old has a much better ability to remember things than you do as their parent. But usually we don't start really having bad memory loss until we're quite old, in the 80s, 90s, if we live that long. But somebody with Alzheimer's, which is also known as presenile dementia, meaning it occurs early, may start having noticeable cognitive loss after the age of 60. It gets worse. It gets precipitously worse as you get older. But the changes in the brain that cause Alzheimer's start many years before it shows up as something that can be diagnosed, which is definitely a big problem with treating it. So years ago, when I first started in this field, there were a bunch of medications that had been approved for treatment of Alzheimer's disease. These all fit in the category of what I call cognitive boosters. So they kind of enhance the ability of neurons to fire and for their signals to be picked up in a very general way. It had no effect on disease process at all. The effectiveness of them lasted a fairly short amount of time and was minor, but it was all that there was. A lot of efforts started being put into what are the causes of Alzheimer's disease, and you can go all the way back to 125 years ago. A German psychiatrist named Aloy, I always forget how to pronounce his name, Aloy Alzheimer, had a patient that he described as having a case of presenile dementia. She was a 51-year-old woman. She was suffering short-term memory loss. He followed her up, keeping a lot of notes on her, and the next slide. Okay. And she died in 1906, and he studied her brain. And this is actually one of the original microscope slides of her brain. And what he noticed were things that did not occur in normal brains, were these, what he described as plaques, so these dark areas, and what he called neurofibrillary tangles. So these very dark, small black spots were actually clumps of dead neurons that were all tangled up. So this is what was known for many years, and nobody really knew what these things were until 1984. The biochemical techniques had progressed to the point where somebody was able to take an Alzheimer's brain, scrape up some of those plaques, and analyze it. And what they found is that they contained a small protein, which was called amyloid beta, A beta. I might refer to it as beta amyloid too. People kind of mix up the names a lot, but it's all the same thing. It accumulates in the brains of Alzheimer's patients. So this is a small fragment of a larger protein that exists in the surface of neurons and other cells in the brain. And that's depicted on the cartoon in the bottom. So you see this protein APP, which just means amyloid precursor protein. Not a lot is known about what its actual function is, but it probably has some function, normal function, since it's there. So that APP is there, and it is clipped by two enzymes that reside in and around the membrane, and these are called secretases. So there's a base or beta secretase and gamma secretase. So these clip APP at different points and release this red protein, which is called the amyloid beta protein or A beta. That small protein goes out, diffuses through the brain. It normally is eventually exported out of the brain and doesn't reach an appreciable level. But in Alzheimer's, this process is speeded up, and you get an accumulation of that A beta protein. When that A beta protein accumulates, a few things happen. It naturally has a propensity to stick to itself, to other molecules of itself. And this can go in two different ways. It can form what's called insoluble amyloid plaques. So it can form really big sheets of this protein, which deposit themselves on the brain tissue. It can also form soluble oligomers. Oligo meaning many. So these are many copies of this A beta protein that stick together. These exist in many different sizes, from just 2 to 3, 4, 5 to 45, whatever. These have effects on the brain. Yeah.
Does the same thing happen other sites outside the brain? In other words, is insoluble proteins occur in the heart, in the kidney, in the liver, in the muscle, or is it restricted to the
I
brain?
think the thing about the brain is that it's kind of a closed system, so you have a problem with it accumulating and not being cleared out of the body. So I think it is produced in other cells, but it doesn't accumulate to the point where it becomes a problem. There are other proteins that I'm sure you know of that kind of accumulate inappropriately in different parts of the body. They might be damaged and they fold up and form clumps. So the word amyloid is kind of a general term. You could have it's kind of a descriptive term, right? And you can have there are other diseases that are caused by accumulation of what we call misfolded proteins. They don't fold up the way they're supposed to. But thank you.
Double binding, not only do they make them, can't get rid of it.
Right.
Might be.
But this has no normal function as protein. So-
Well, does it have a normal function? That's a good question, and there's a lot of research that's been put into that. Both the single A beta peptide itself, the clumps of it. I don't know if that has too much of an effect, but and the whole APP protein and the fragments of it. I mean, people in experimental systems can look at functions of these things and they find a lot of functions, but it's really unclear what's actually going on in the brain. But I'll touch a little bit on what is thought to happen sometimes when some of these are blocked, because some of these components may have beneficial properties in terms of keeping our brain connections tuned up. So nobody really knows what triggers this excessive accumulation of the A beta protein in most people. But it is known, so it's another important piece of data, as people started developing techniques to look at how gene variations are related to diseases. And it was found that the strongest genetic risk factor for Alzheimer's are variants of a protein called apolipoprotein E, which for obvious reasons is shortened to called ApoE. So ApoE has a lot of functions in the body. It's involved in transporting cholesterol. It's kind of a transport protein in the blood. In the brain, it appears to also be involved in taking this A beta protein and going to the blood-brain barrier and shipping it out of the brain into the cerebral spinal fluid, which then drains into our blood system and gets cleared out of the body. So that's its normal role. But it's found that there are the three main variants of this ApoE, ApoE2, ApoE3, and ApoE4, have different abilities to clear A beta. Most people have 2 copies of ApoE3. But if you have 1 copy of ApoE4, ApoE4 is less effective in clearing out A beta. So you get an exaggerated accumulation of A beta in the brain. If you have 1 copy of ApoE4, you have double the risk of getting AD as the people who have 2 copies of ApoE3. If you have 2 copies of ApoE4, you have 10 times the risk of getting Alzheimer's disease. So it's a very important risk factor. There's also an ApoE2, which is present in a much smaller percentage in the population, and that actually is more effective in transporting A beta. And those people who carry that, 1 or 2, there are very few people who have 2 copies, but if you have 2 copies, you actually have a much lower incidence of getting Alzheimer's. So because of these things, because of the finding of the plaques and having the A beta protein in there and this genetic linkage to handling of A beta protein, most of the therapeutics over the past 20, 25 years have focused on what's called the amyloid hypothesis, and has focused on 2 main therapeutic areas, amongst others, are inhibiting those secretase enzymes that are involved in clipping that precursor protein and making the A beta, and also on antibody therapies that will bind to A beta and clear it out of the brain. So very intensive research and drug development in these areas, and this led to basically a dismal decade of clinical trials. So I'm not going to talk about all these things, but you can see they are represented by the biggest pharmaceutical countries in the world who are involved in this, who spend billions of dollars on clinical development. They're in different categories of things. So there are 2 different secretase enzymes. There are inhibitors for each of these. And there are antibodies to A beta that are used that were tried in clinical trials, and they all failed for the main reasons being excessive toxicities, especially with these secretase inhibitors, trying to make them selective enough to inhibit these secretases and not inhibit other enzymes, cause toxicities in the liver and other organs. Some of the secretases especially, actually worsened cognition. That led to the idea that, well, maybe this A beta protein by itself is important. That maybe it's important for actually having healthy neurons. We don't want too much of it, but having a right amount is important. And the antibodies just generally were not improving cognition or slowing the loss, the decline of cognition. So this was pretty devastating in the field. It really, one trial after another, and companies spending billions of dollars on some of these trials, just led most of the big pharmaceutical companies to back away from Alzheimer's research and development. And some, including Pfizer, totally got out of the whole field of CNS pharmacology. They're kind of sneaking back into it now in kind of a backdoor way. So it was very, very hard. Now, I was working through this decade, I was working for this startup company. It made it very hard for us to get investment money because people are like, Alzheimer's can't be cured. Why are you trying to do this? And we were dumb. We kept trying to work on it. We didn't have stockholders most of the time either, so we could do it. But finally, this kind of started changing in 2021. The first antibody treatment for clearing out A beta plaques was approved. So it was a monoclonal antibody made by developed by Biogen. It was shown to slow disease progression, so slow the rate of cognitive loss. Now that statement right there is actually very controversial. And the approval of Aduhelm or aducanumab was very controversial at the time. They did 2 trials. In one trial, they saw a significant slowing of cognitive decline, although there was a fair amount of skepticism about the statistics they used and how they loaded patients in the trial. And then did another trial where there was no effect. And if you combined all the patients from both trials, the statistical significance you saw in the one trial got washed out. So I listened in on the FDA advisory meeting, and they as part of the process, the company presents their data, the FDA presents their version of how they interpret the data, and it was like amazing because it was like they were giving a company presentation. The lead investigator at the FDA was all in. And then there's always an external independent review board. And I believe there were 12 external reviewers. So these are all scientists in the area and clinicians and researchers. They're not tied to the company at all, and they give their recommendation. And out of 12, 11 of them said this drug should not be approved. It's not effective. The 12th one said, I'm not sure. So yeah, so this is an advisory panel, and the FDA can either follow their advice or ignore it. I think it was significant that one of the people, because they also, at these meetings, they also have testimonies given by patient advocacy groups, public citizens always there, usually saying, this is toxic, we shouldn't be giving it to you. But there was somebody, and I forget who it was, they said, yeah, this is probably not the best drug in the world. I'm sure there are ones that will come along that will be better than this, but if this drug isn't approved, those drugs will never come before us because everybody's going to get out of the field. So the drug was approved. Okay. I'll talk a little bit about it. Yeah. So this was approved in 2024. It was actually discontinued it was approved in 2021. It was discontinued 3 years later because the company that was putting it out, Biogen, had another antibody therapy that they had been worked a little bit differently, that they were developing it alongside, and it looked like it was going to be have a better effect than the first one that they had. So they discontinued the first one and focused on the second one. And then shortly after that, Eli Lilly, they had also stayed in the game with their antibody therapy, and that was proved. So when I say it was effective, what does that mean? So a lot of stuff on this slide here. But if you look at panel B, which is in the lower left there, that shows the amount of amyloid plaque in the brain. So these antibodies were designed to clear A beta out of the brain, and early studies showed that they did that and actually reduced the amount of amyloid plaque. You can measure amyloid plaque in brains by doing a PET scan with a particular agent that's given to the people, an agent that was first developed here in Pittsburgh. It's called Pittsburgh B. There are other ones that have come along that I think work a little better than that. And you can see the blue line is some patients on placebo, and the amount of amyloid burden in the brain stays unchanged. And people who are on the drug, you can see with the yellow line, you could see a progressive decrease in the amount of amyloid in the brain. So they're doing what it did, but there were other antibody therapies and secretase therapies that did a great job of lowering amyloid plaque, but didn't have a significant effect on cognitive scores. But this, you can see the graph at the top there shows one of the main cognitive scores that are looked at. And here you see the blue line again is placebo, and you can see the worsening cognitive score over time. This is 18 months in this study. And then lecanemab, which is the yellow line, you can see there's a slower decrease in the cognitive scores. And the lower right one is just a different measure. They have a number of different measures they use for cognition. So this was statistically significant, but you can see it's slowing the progression, but it's not stopping, right? It's maybe slowing it by 6 months, but the disease keeps going on. So this is, hopeful, but somewhat disappointing because, it's still not something that's stopping the disease. Now, you might notice here that the graph, the showing the amyloid PET goes down to about 50% of where you started. Maybe they're not clearing enough A beta out, but if you push these antibody concentrations too high, you start getting a problem that's known as ARIA, which doesn't have anything to do with operas. It stands for amyloid-related imaging artifact. How do you like that for a medical term, right? So this is just something that shows up on MRIs of the brain, and you can see the arrows pointing to these white areas. It's a buildup of fluid in the brain. At higher concentrations of those antibody therapies, you get more of this. It's related. It's not fully understood, but it's believed to development of leakiness in the blood vessels in the brain. It occurred in the approved drugs at the approved doses. It occurs in 10 to 25% of the patients. It's worse in patients who have that ApoE4, which is pretty highly represented in the population of people with Alzheimer's disease. Most times it's non-symptomatic. When it does have symptoms, they include dizziness, nausea, headache, and if it's really bad, you can have seizures that are associated with it. So this was, it was decided that since most cases are not symptomatic, that it's acceptable. But if you go on one of these drugs, you get monitored with follow-up scans to see if this is developing. And if it does, then, I'm not sure what they might have to take you off the drug. They might have to give you a holiday from it. It's like a lot of therapies. You have to manage what the symptoms are and hope they don't get worse. So plus there normal reactions you would expect of an antibody therapy, allergies and things like that, injection site soreness and everything like that. But this is kind of the most troublesome one because nobody's really found a way to curtail this. Ironically, one of the problems with giving antibodies to people is that for treating a brain disease is that antibodies don't get into the brain very well. I know people who are I've worked with people who are very adamant saying they don't get into the brain, but a little bit gets into the brain and enough they're actually clearing the A beta. Some people think that this leakiness of the blood vessels is part of how they get into the brain and clear out the A beta. So it's definitely a two-edged sword. But the risks of these therapies and the other thing about trials that we use to approve is that at some point people thought, well, maybe we're treating people too late in the disease. That once you get to a certain point, people have lost too many neurons, there's too much damage in the brain, and you're not going to reverse that. So let's treat people earlier. And those trials that were all approved it was approved for people with early AD or what's called, and also what's called mild cognitive impairment. So that's what you had to do. So the fact that now you got to watch out for this side effect here. It's only good in people with it's only approved for people with very mild cognitive loss, has really limited the number of people who have been prescribed this drug. I wasn't able to get current numbers, but it's probably in the hundreds of thousands. At first, when these were approved, since most Alzheimer's patients are elderly and are on Medicaid, people were thinking this is going to totally blow out all the costs of Medicaid and Medicare. It would, that I saw predictions that it would, maybe increase Medicare premiums by 50% just to cover this treatment. But that's been limited by the fact that they're not really prescribed in too many people. Yeah.
How effective is it?
How effective is it?
Yeah.
Well, like I showed it slows cognitive loss, but it's not stopping the disease. Yeah.
For some time, starting from winter in late April.
Right. So that shouldn't be there. What's going on now? So one of the things is because these drugs work better in early cases of Alzheimer's, a lot of work is currently going into earlier diagnosis and intervening earlier. So there are a lot of brain imaging techniques that are being defined and improved upon. And there are what are called biomarkers. These are little fragments of protein. A lot of times they're residual parts of the neurons that are dying in the brain. There are measurements for these A beta proteins, and it's a whole collection of different proteins that come from the brain, and their concentrations are changed during the course of Alzheimer's disease. They get cleared out of the brain into the CSF, so you can measure them by drawing CSF and doing various tests to measure them. That's not that easy to do, so there's now a lot of effort put into the detection of these proteins when they work their way into the blood. So a lot of effort being put into simple blood tests that could be done. One of the other things is that if you look at the data from people at various stages of normal health and stages of Alzheimer's, the values of these markers are all over the place. So it just varies a lot. So what's really important is looking at people over time and tracking it. So a simple blood test might not be the answer, but okay, we'll test you now. We'll test you a year from now. We'll test you 2 years, like that. So how do you do that? So there have been a lot of long-term studies going on in populations of Alzheimer's patients and also in normal people. So some of the biggest ones are called ADNI, and University of Pittsburgh is a participant. in this program where they track people. They track people with cognitive loss. They track normal people too. They want to see, and they're collecting blood samples from these people. They're doing brain imaging at periodic times, and they want to look, how are these factors changing, and when, at what point do people start developing cognitive loss, if they do at all? There's another program called the Dominantly Inherited Alzheimer's Network, or DIAN. This is for people who have those variants of those genes that cause a genetic propensity. So they specifically look at those people. If they can identify them and say, well, there's a much higher likelihood that these people, especially if you have 2 copies of that ApoE4, maybe these people should be put on a therapy before they start developing symptoms of the disease. And another interesting one is called the Religious Orders Study. So this is a study of nuns who are living in communities. Benefit of these, they have a very similar environmental background. They're all living together, eating similar diets and everything like that. And you can track them over decades. So there are a lot of nuns who have signed up to be part of the study, and they're being tracked over time and see which ones of them develop dementia or not, and how that can be matched up with some of the markers that they're looking at.
How do you diagnose this IPO or 2?
How do you
How do you test, know that people have that or not?
if they have Alzheimer's?
No, if they have those genes, the propensity to.
So it's like any other genetic test. You can take take a swab of somebody and sequence either specifically those genes or.
People who are concerned, maybe their parents or grandparents had Alzheimer's, they can elect to have those genes.
You can. And it's something that you should talk to your physician about, because there are pluses and minuses to that. Some people think, if I'm going to get a disease, maybe I don't want to know about it, because especially if there's no good treatment for it. Other people say, well, if I know I'm going to get the disease, I want to do things now that, rather than putting it off. So it's something that the decision that people have to make, and you should talk to your physician if it's something you're concerned.
Gene therapy a possible route, either to block APO4 or promote APOE2 production?
Sure, it's a possibility. Doing gene therapy in the brain is very difficult, but it is something that is being looked at early stages now, as well as cellular therapies, putting in precursor cells that might create new neurons. This is kind of an overhead view of the current therapeutic pipeline for Alzheimer's disease. So there's this Dr. Jeffrey Cummings. Every year, as long as I've been in the field, he's done an update of this article, and they have very thorough ways of searching and looking at things that are being developed. So you could pull out all his articles every year and see how this is changing. I'll just point out there are, this chart is in different, is organized in a specific way, where you have the beige slice of pie are cognitive enhancers that work like the old Alzheimer's drugs. They're always trying to find new ways of doing that, just trying to boost the function of the brain cells that are still there. There are neuropsychiatric symptoms. So some of the things like, what's that?
These ads for Prevagen.
Right.
TV and-
Yeah. Show me the clinical trial for Prevagen, and I'll
Yeah.
At home to repeat the question.
Okay. Yeah. So Mike asked about Prevagen, which is something you see a lot of ads on TV for, and I won't make any comment other than. Yeah, I did make a comment. Yes, I did. I'll make a comment by not making further comment. There mentioned certain like neuropsychiatric problems, depression, insomnia, confusion. So there are drugs that are out there for those things. And sometimes they're actually just testing those to see if they work in Alzheimer's patients who have those symptoms. And there are some other, maybe more specific things to Alzheimer's disease. So that represents part of this pie. And then the big part of the pie, which is about 2/3 of it, are therapies that are designed to target the disease process. So these include biologics like antibodies and some other types of growth factors and things like that in the top part of the pie. And then the big purple area, which are small molecules. So these are things that can often be taken as a pill. You also see there are concentric circles here, and this represents a different phase of drug discovery, of clinical trials. So the outer ring are phase 1 trials. So this is the first studies in people where you're looking for the safety profile of the drug. The next phase are phase 2, where you're trying to look for whether the drug has some activity that is shows that it might be working, but they're usually smaller trials, less expensive trials. It helps you figure out which dose you might want to use in a bigger trial, how long you need to treat. And then the inner circle is our phase 3 trial. So these are usually bigger trials, more expensive trials, probably longer duration, that are designed to generate data that could be used to present to the FDA to get approval. Now, normally, it doesn't, this article, I looked to see if it mentioned, normally there are a lot more drugs in phase 1, because a lot of things, it's easy to get a drug into the early studies. Most things that go into phase 1, or a lot of things that go into phase 1, are eliminated because there's some excessive toxicity. The phase 1 circle isn't bigger. There aren't more drugs in that than the other ones. And I don't know if that's just because drugs usually don't spend as long in phase 1, or because there aren't new things being developed. I don't know, but it was just an observation I made. So looking at all of these things here, I just want to draw your attention to this one little thing here that popped up in red. It says CT-1812. This is the drug that I worked on for a good dozen years. So it's in there. It's in phase 2 studies. I've got a little time. I can go very closely. So this is still affecting the amyloid beta pathway, but at a different point. Instead of looking at the plaques, those insoluble deposits, we looked for something that would affect those soluble oligomers. So over 10 years of research, we've kind of gathered a lot of data, and this is kind of a model that we came up with, which is not a proven pathway of how it's working, but I'll say it's consistent with the data that we've gathered in our lab and with collaboration with a lot of other labs around the world. So in the panel on the left, this represents part of the surface of a neuron in a synapse. So synapses are where connections are made between neurons. It's kind of where the action going on, how neurons communicate with each other. So there are a bunch of proteins out there. They kind of float around in the membrane. Some of them move in and out of the membrane. They all have different normal functions. But at some point, they do kind of stick together, come together for a little bit as part of their normal function. But what we've seen evidence for is that in Alzheimer's disease, these A beta oligomers bind to specific points on neurons in the synapse. And we think what it does is it causes some of these proteins to clump together and stay together in the surface of the cell. So they can't do what they're normally supposed to do in their normal processes in helping neurons function properly. And that we screened for drugs that interfered with the sticking of A beta to neurons. And we found compounds that were active in this particular 1812, which was our clinical turned out to be our clinical candidate. That's on the surface of the cell changes that protein enough so that the proteins that are binding A beta kind of come apart and release A beta. So this, when A beta oligomers are in the brain, it causes synaptic damage and loss and memory failure. Kicking off A beta, the theory is that it restores synapses, which is something that we can see in artificial setting where we can kind of reproduce Alzheimer's disease on cells in a culture dish. restores synapses and hopefully restores memory. Memory. So right now, drug has a name now, zervimazine, which I'm still learning how to say. So we discovered it in 2014. In 2017, it initiated phase 1 clinical trials and got through that fine. Since then, there have been a number of phase 2 studies going on. Again, like I said, it's trying to find a dose that works, that kind of limits what some of the side effects are. Just mention some of them. So the one that's really cool is that we did an acute study where we just give like one dose of the drug to somebody with Alzheimer's disease, and then we pulled cerebral spinal fluid out at 1 hour intervals over 30 hours. And what we could see was after you gave people the drug, and we had to develop methods of detecting amyloid protein in these oligomers in very small quantities, and we could see it start appearing in the blood over that first 24-hour period. So this was pretty remarkable.
How do you introduce the drug?
What's that?
How do you introduce
The drug is given as a pill. It's an oral pill that's given once a day. There's been a 6-month study in 120 patients, which is pretty small by Alzheimer's standards, and this showed trends towards cognitive loss. It also showed improvement in some of those protein biomarkers that you can measure in the CSF. There was another 6-month study that showed that certain areas of the brain that are involved in cognitive function, which are known to atrophy during the course of Alzheimer's disease, that we saw kind of a stop of that volumetric loss. There was another study, it was just a 4-month study. It turns out that there are ways of looking at brain waves, so EKG waves, that there's a way of analyzing EKG waves to tell whether different areas of the brain are connecting with each other. So this is important. This is like our neural pathways connect different areas of the brain. In Alzheimer's, it was found that this particular approach showed a decrease in communication by different areas of the brain. And the study that we did with our drug showed that this connectivity was restored in a pretty short time, which was pretty remarkable. So currently it's in an 18-month-long trial in 540 patients in about 40 different clinical centers around the country, including here at the University of Pittsburgh. That's a study that's heavily funded by the NIH. So thankfully, they haven't cut Alzheimer's research yet. The whole time I was working in it, we got a lot of SBIR grants, which are small business grants for businesses. Well, obviously, every year I was working in the field. The Congress voted an additional $300 million to put into Alzheimer's related research, which say, well, that's one of the benefits of having a Congress full of such programs were stagnant or getting cut. They kept putting more and more research into Alzheimer's. So anyway, it's actually the largest clinical trial study that has ever been funded by the NIH is this study, the most expensive one anyway. The company has also done studies in a different disease, dementia with Lewy bodies, which is another disease that involves cognitive loss. It's a little bit different than Alzheimer's. It's kind of like a cross between Parkinson's disease and Alzheimer's disease. It's actually a disease that yeah, I'm losing my memory. I worked for 10 years with these peptides that cause Alzheimer's disease. Mork and Mindy.
Robin Williams.
Thank you. Robin Williams was diagnosed with dementia and Lewy bodies, and he took his own life actually. So anyway, and so there's been one trial in there, and it's thought that it had some beneficial effects there with cognitive loss, and also kind of a bit different than this. we had reason to believe that it might be effective in protecting the retinal cells in the eye, because there's some similarities actually to what goes on with aging of the eye and Alzheimer's disease. And so it was put into a small trial of age-related macular degeneration. So this is it's actually one of the biggest causes of blindness in the world, and it showed some effectiveness there. So anyway, that's different areas where this can work. Okay, so the therapeutic picture is not great. There continues to be a lot of work. Oh, I should have mentioned that these disease targeting therapies involve a lot of different mechanisms that have come up in the research over the years. So people are trying a lot of things. It's thought maybe you need to have more than one drug combined together, but you first have to have those drugs approved before you can start. So there's some rational thought behind, okay, this drug's approved and this drug works by a different mechanism. Maybe eventually we can combine them together and do things. Especially in the area of inflammation. So inflammatory processes of the brain are not as well understood as they are in the periphery, but there's definitely an inflammatory component to Alzheimer's. So what can we do? So there are a number of lifestyle things that people can do to help decrease their chance of getting Alzheimer's and other dementias. One of the biggest ones is controlling high blood pressure. So if you have high blood pressure, getting it treated, if you're getting it down to your systolics, that's the higher numbers, systolic and diastolic numbers in your blood pressure, getting the systolic pressure below 140 has shown to have about a 10% lower chance of getting Alzheimer's disease. Getting it down below 120 has an even bigger effect. Depending on the study done, it can decrease your chance of getting Alzheimer's by as much as 25%. So that's a pretty good thing. Managing blood sugar, this might have to do with some of the inflammatory processes in the brain. It's not quite understood, but if you have hyperglycemia or something, it's something you should talk to your doctor. Blood pressure is very controllable. Something you should do it, and the earlier you do it, the better too. So don't wait. And then a lot of things which are generally healthy things that should be doing anything, maintaining healthy weight, having a healthy diet. And it's interesting. So I got this off of Alzheimer's.gov like a couple of days ago. And my first thought is like, has this been altered? So it still says limit fats and sugar. And considering it doesn't say eat more steak and I haven't mucked with these recommendations yet. Keeping physically active has a big component. I know Richard kind of did a lot of work with neurodegenerative diseases and physical activity. No, am I wrong there? No. Okay. Okay.
I did put something.
Okay.
Quitting tobacco.
Use, staying mentally active. You've probably heard that a lot, and all these things are good things to do anyway. Probably staying socially connected, I thought was an interesting one too. That seemed to have a beneficial effect, as well as treating hearing problems. That's kind of an odd one you wouldn't normally think of, and partially it affects our ability to be socially connected to people. If we having trouble conversing, it tends to make people isolated. Limiting alcohol use. Severe alcohol use can cause damage to the brain on its own, and getting a good night's sleep. So anyway, that's kind of where we are. Yeah.
Are experiencing normal cognitive decline with age.
Yeah.
You're having a problem that we should deal with.
Hard one to tell, but I would say if you think you're having problems, or if people are telling you, it's like, gee, you don't remember things, you should talk to your doctor about it. Maddie, do you have a comment about that? Because I know you give Maddie the speaker there.
we all have this A beta in our brains, right?
Right.
What are the current theories about why we have it? Are there any current theories about that?
We have it. Yeah, there are a lot of theories that it is actually involved in the synaptic connections. So the neurons in our brain are always reaching out and making connections to other brains. If all those connections became permanent, we wouldn't be able to keep anything straight. Our thoughts would be just crazy. So there's a normal process of creating these little, they call it synaptic spines, these that come out from neurons to touch other neurons. So there's a normal process of those being made and then being trimmed back. So when something happens and your synapses are connected, if you think about it, if you remember it, if you're reminded of it, it kind of makes that memory permanent. Those connections become permanent. There are biochemical changes that go on in the neurons that make it permanent. But most of those synaptic connections are trimmed back because it's not important. So they start to be involved in that normal process. It's part of the reason, one of the hypotheses why those secretase inhibitors, some of them cause cognitive learning, worsening, and that the A beta protein might have some beneficial effect in terms of keeping those connections healthy, and that by cutting it down too much, you're actually interfering with that healthy process. So it's kind of a new generation of secretase inhibitors that kind of are less effective. They don't really shut it down all the way. They allow some clipping of that protein, but not as much. And those are in the early stages of research. I'm sorry to go back. Yeah, I would talk to your physician if you think you're having a problem. They would do cognitive tests on you. If they think it's really bad or they think it's worsening, there are brain imaging that can be done, and they look for things like that buildup of that plaque. Although a lot of people have a head full of amyloid plaque, they have no problems with their cognitive abilities. They die with a head full of plaque still being unaffected. So it's the connection between that and the cognitive loss is really.
Do we still need to screen share do we still need to screen share?
I just wanted to leave. So this is just a list of online resources that you might want to look at. And if you want a copy of these, I can send these to you. But that's other than that, no, I don't think we need to screen share.
Am I able to say something remotely? Maybe not.
Sign the,
Good idea.
that went around this.
We've got another 10, 15 minutes for questions.
Are you aware of any studies of twins?
Twins?
Yeah.
Well, not particularly, although there are a lot of family studies. Oh, the question Carol asked if there were any studies of twins. So there are a lot of studies on the genetics of Alzheimer's disease that I'm sure took into account twins and other family members. But I don't know specifically of studies of twins, although if one person if you have a sibling, any sibling who gets Alzheimer's disease, that kind of doubles your chance of getting your probability of getting Alzheimer's disease. If a twin gets it, I don't know. Well, my dad had a fraternal twin brother, so he got Alzheimer's disease in his 60s, and he passed away about 10 years ago from it. And my dad is 90, and he is sharp as a tack. So I don't know. There are also communities where there are a lot of closely related people. So in particular, in Medellin, Colombia, there are family clusters where they have these A beta, these bad A beta genes, plus some other genetic traits that predispose you. And if you're in one of these families, there's a 100% chance you're going to get Alzheimer's, and you might get it even in your 40s. Okay. So there's a lot of studies being done on this Medellin cohort. There's populations in Iceland, families in Iceland, where they hardly ever get Alzheimer's disease. And there have been some genetic variants found in those people that seem to protect you against Alzheimer's. They're not only against Alzheimer's, they protect you against all forms of dementia. So, that's kind of giving clues into Down syndrome. Yeah, so Down syndrome, it's interesting because this APP protein is the gene for it is located on the chromosome that is duplicated in Down syndrome. So, yeah, right. So you have 3 copies of chromosome 21. And people with Down syndrome do have an excess amount of A beta protein and plaquets build up in their brains. And, I know some guy in the field who he worked with C. elegans, which is a little worm model. And he had, his son was born with Down syndrome, and he got interested in Alzheimer's disease. He created a model of creating Alzheimer's in the thing that he knew, these C. elegans proteins, because he had an interest in the son. And the company where I worked, we actually had a guy work for us part-time. He's a biochemist at the University of Pittsburgh, has a son with Downs, and he was drawn to the research because of his son. But whether it works particularly well in people Downs, it's hard to start a clinical trial in young children. So maybe if there are things approved, they might use it. Yeah, Richard.
Well, there's some of us in the neuroscience community that are really skeptical of the A beta hypothesis at all. Of course, you just mentioned some people die of A beta of the brain.
Right.
Mind. One of the things in this people has a 2q4 allele. The first place the A beta shows up in the brain is actually the basal ganglia, and that does not stop functioning. So it's a little bit uncertain.
Yeah, definitely. Yeah.
In about 2000 to 2004, there was a company, I believe it was Elan, that immunized people with A beta. So they naturally produced antibodies to A beta. And they had to stop that trial because of the similar things that these other antibodies are causing. There's some brain swelling, and I think several people died. But 5 of those people eventually died, and they looked at their brains, and all the A beta was gone. They're completely clean. Had no effect on their cognitive decline. Now, the only problem was I think, were moderately impaired when they started the trial. So at some point, it seems irretrievable. So there's also a lot of interest in the tau protein, which makes what's called neurofibrillary tangles within axons. And the distribution of tau in Alzheimer's patients seems to better mimic the deficits that are seen early in cognitive decline.
Yeah, I didn't have time to talk about tau at all.
I know that's a whole another discussion.
So certainly, yeah. I mean, you bring up a point which kind of is why I worked at a small pharmaceutical startup that's still a small pharmaceutical startup. It was always impossible to draw financial interest from big pharmaceutical companies because as kind of legs were being kicked out from the amyloid hypothesis, we kept saying, yeah, but this isn't plaque. These are oligomers, and we know that has effect on these synaptic connections. Maybe it's different. And the company went public in 2021, and their share price is hovering around $1 a share right now, which is keep getting threatened to be delisted. So it's definitely an uphill battle. So yeah.
Is there another alternative explanation for why APO4 is strongly correlated with
1804 was not active. There was an 1804, but 1812.
APO4?
Oh, APO4. I'm sorry. I thought you said 18. Okay, yeah.
Is there an explanation that doesn't involve this A beta?
Yeah.
For why APO4 is bad and APO2 is good?
So APO4 does other things
Okay.
too. And one of the, kind of new focus is on cholesterol and how our brains need a lot of cholesterol. So there are a lot of things that go on in the peripheral parts of the body that also go on in the brain, but how they work in the brain isn't as well studied. It's kind of hard to study the brain, but yeah, apolipoprotein E has a lot of different functions, and yeah, there are definitely other things that could be related.
Can you say something about blood tests availability and their suitability?
I don't know how available they are, actually. They're still, or I don't know how commonly they're used. As far as I know, they're still kind of in an experimental phase right now. And with any blood tests, it's only as good as understanding what the predictive value of them are. So, right now they're trying to understand what do the changes mean and how do they relate to, is this actually disease or is this not disease, and which ones are the most important ones. But
And I've only written one paper on Alzheimer's in my career, and it was about the inverse relationship between cancer and Alzheimer's with somebody here at Pitt, Mary Ganguly. And apparently they're having a big conference at the NCI to look at that. I don't know if that's something that's come up.
I don't know. I'm not really familiar with that work at all, but that's interesting.
So
If you have, the good news is getting Alzheimer's, you have less risk of getting cancer and vice versa. So you can look at it however you want.
There's a terrible joke about that. I won't say that. Yeah, Marty.
Is work being done on other forms of dementia? I mean, are there similarities? I know my mother had vascular dementia, which is a completely different mechanism.
Right, yeah. Vascular dementia is, I think, maybe the second leading cause of dementia. You need blood flow, right? Your neurons need oxygen. You need good blood flow to clear out waste from your brain too. Yeah, it's something that I've never gotten into or looked into that close. Every once in a while, the CEO of my company would say, my original background was in cardiovascular sciences and therapeutics, and so you should look into that vascular. I said, I've got too much stuff to do with what we're doing here. I can't.
So none of this affects the other form.
But that's why, so certainly controlling your blood pressure, your systemic blood pressure is pretty important. So if high blood pressure, it puts a lot of stress on blood vessels and capillaries. You can get breaking down of it, and that's not good. So.
I see Ali on the call. Thanks. So anyway, say that there were symptoms of Alzheimer's in the president.
is there a drug for trunk derangement?
I am not a physician or a psychiatrist, so I can't diagnose. I would say he's not a paragon of mental stability or cognitive function. I would definitely feel semi-qualified to say that as a citizen of the world.
Can I ask a question online?
12, maybe one more question.
Can I ask a question online?
I think Bob is trying to say something online.
Can you hear me all right? Can you hear me all right?
Bob, you may be muted.
No, I'm not muted.
You're still muted.
I can hear it because I got a speaker here, but it's very faint. Bob, if you're Bob Mitchell, if you're
Check and see what speech it's using. Hold on.
oh.
So can you hear me better now? Maybe if I got closer.
Can you type it in the chat, Bob? Can you phone it in?
Okay.
He's on his laptop. He's been.
Okay.
You must be trying to type.
Well, I just want to thank everybody for coming this morning. Feel free to ask.
You comment on the 2006 paper that had fake data.
What?
Fake data.
I'm not sure which fake data you're talking about. Unfortunately, that's kind of a broad question. I mean, there have been a number of papers that have been retracted and you know.
Is it true, Nick, that if you, I think there's research it says that if you sip single malt scotch, that it improves brain function?
So the question is, if you sip single malt scotch, does it improve brain?
Yes.
Yeah, well, I will say that I had the good fortune of being able to have dinner in Aberdeen, Scotland, or actually in Edinburgh, Scotland, with one of the foremost researchers in Alzheimer's, and we did drink a fair share of single malt scotch. If he's all for that, then I think it's probably it's not a bad thing.
Light applause.
All interested in this.
I know.